Now we're going to look at another real-world application of this technology. Uh, in specifics, we're going to look at paternity testing. You could also use this technique that I'm going to show you to identify DNA from an individual at a crime scene and match it to that person's DNA sequence. What I've done here is drawn out an example chromosome for each of these individuals. Keep in mind, if we're looking at, say, chromosome number 4 for this woman, then uh, you're also going to have another chromosome here, homologous chromosome, right? Because she's going to get one of these chromosomes from her father, one from her mother. To avoid complication, I'm simply going to just display one. These dots that are all over the place are going to help me to cut these chromosomes. So we'll do that later. The child, however, I've drawn two because it's sort of important that you understand what's happening here. Two chromosomes. Okay, so let's say we take some enzymes that cut DNA in very specific places. They recognize specific sequences. So we digest or break up the DNA using that enzyme. And for the woman, let's say we have cuts here, here, and here. That gives us now, instead of one long chromosome, we have 4 segments. And if we run that through a gel, we should get 4 bands. And so that's what we've gotten here. We've gotten 4 bands that have separated based on size. The longer segment here corresponds to this band. The shorter segments here correspond to these bands that are close together because they're relatively similar in size. For the males, we're going to use the same enzyme cutting at the same sequence, but that sequence may be located in different spots on their chromosome simply because when you have a chromosome, there are certain portions of the chromosome that in some people are longer and some people are shorter. So the sequences might be a very long sequence, making this really long fragment here, where here's the place where the enzyme cuts, and yet there's this huge spot in the middle. Sometimes there's a mutation where you don't have the place where you can cut because the enzyme doesn't recognize mutated sequence. And so in this individual, for example, we only have 3 segments created instead of 4 because they're missing that spot. So there's a lot of variation in humans. And when we do this type of testing, we use an enzyme that will cut in a lot of places. It'll cut in dozens of places so that we can get what's called a DNA fingerprint. Now we look at the child. And the child's DNA, we can cut using that same enzyme. It's going to cut at the sequence it recognizes. And this is what we get. Now let's go over to the gel and see what we have. In spot 1, we have, and I've just drawn the symbol for female, so we have the woman, and the symbol for male here for these two. We have 2 males, and we're asking the question, which of these 2 is the parent, biological parent, of this child? And we can do that by the process of elimination, essentially. We know that any stretches of DNA that this child has is going to correspond to the DNA that their parents gave that child. And so we should see the lengths of fragments in the child match up fairly similarly to the lengths of DNA we cut up in their biological parents. To actually do this, let's look at the mother first. And so the mother has these 4 segments, and we can go over to the sample 4 and say, okay, where do those segments appear in the offspring? And let's just cross them out. So this is the mother's contribution to chromosome, let's just say chromosome 4. Here's another contribution. and then we can get rid of these two. So we're left with a segment here, and a third segment here. Now it's pretty straightforward. What we're doing is trying to find the other individual whose fingerprint matches these fragments. They do not come from the mother, they have to come from the father. And so I made it easy, just drawn it right here. We know that segment is the same as this one because it's on the same length through the gel. This is the same, and this is the same. So now we have determined that the biological father is the second male listed here, person number 3. Fun stuff.